Hi, I'm Dan Heckman. I'm a Durham resident at the Medical College of Wisconsin, and I will be giving you your pathology lecture today. Really, you look at it, the pathology section is a pretty small section in the first aid book, but actually it's really, really important. Pathology in general, or questions related to pathology on your USMLEs, will comprise about 60% of your test. So it's really important that you pay attention and learn this framework and foundation that I'll try to provide you in this chapter that'll serve you well when trying to learn the rest of the pathology sections later in the book. So pay attention, it'll be fun. I'll be really visual and interactive. I'm going to ask you like a million questions. So try to, I know you'll sound stupid, but try to just answer the questions out loud. It'll save you from falling asleep at your computer. And you might even have some fun along the way. So, alright, get ready for an awesome pathology review. Okay, so basically, here's like an overview of what we'll talk about in this section. So, cell injury, inflammation, and neoplasia. So really only three things. We'll start off with talking about cell injury. We'll start off with a flash quiz. So the question is, what are the two main ways cells can die after they've been sufficiently injured? So there's two main ways a cell's been kicked around enough that it can go. What do you think? Well, the answers are apoptosis, which is essentially programmed cell death, and another way to think about it is cell suicide, and necrosis, which is massive cell death. And necrosis in general is pathologic. It's out of the ordinary, and apoptosis is not pathologic as much. It's more physiologic, as we'll see. And we're going to start talking about apoptosis first. Okay, so apoptosis. So again, what is it? We just said that it's essentially programmed cell death, and when a cell wants to die, it basically commits suicide. And kills itself. And so that's what it is. So let's talk about the general mechanism of how it works. So here we have a cell membrane, and this will be like a fun little animation. I love to do animations, and I'm a very visual person, so I hope that's okay with you guys. I hate outlines and I hate words on the screen, so I try to use as little as possible and as many pictures as I can. And things moving around. So here we go. Here's a cell membrane, and there are two main proteins associated with apoptosis, BAX and BCL2. Here's a question for you. Which one do you think of these two proteins is pro-apoptosis? That would be BAX. Which one is anti-apoptosis? That would be BCL2. Duh. Here's a fun little tie-in though. So BCL2 actually is associated with a certain malignancy. Do you know which malignancy it is? It's follicular B-cell lymphoma. And there's actually a mutation with that malignancy. Do you know what that is? So it's a translocation of the chromosomes 14 and 18. And what actually happens is the BCL2 protein is actually thrown onto the heavy chain gene of the antibodies on chromosome 14. And so every time that the B cells make an antibody, they also make a BCL2 protein. So there's just an incredibly increased amount of BCL2. And as we just said, BCL2 is pro or anti-apoptosis. It's anti-apoptosis, so that means that whatever cell has an abundance of BCL2 will not die. And it will hang around and possibly run the risk of turning into cancer because it will just keep on proliferating and not pass away like normal cells do. Okay, so back to our apoptosis. I'm sorry, I go off on little tangents occasionally. I hope that's okay, but I think it does help in general to tie in other concepts and to help you remember things better. It gives your learning more layers. Alright, so yes, the BAX protein will be increased in apoptosis, and the BCL2 is lowered. So when there's an increase in BAX, that will stimulate the release of what from the mitochondria? That's right, cytochrome C. I know you knew that. So cytochrome C will then go and in turn stimulate enzymes that are called Cas-based enzymes. Cas-based enzymes go and degrade a bunch of things. Like, what do you think that they'll take out? So the cell membrane, like I'm pointing to here, they'll take out other membranes like the nuclear membrane, lysosomal membranes, and the lysosomal contents will leak out like enzymes and cause further degradation of things. So eventually everything will be degraded and condensed into one hyaline mass, and even smaller hyaline masses called hyaline bodies. And the word hyaline really essentially, you'll see this over and over again. It means, what color is this? Pink. Okay, it's a fancy term for the word pink. Okay, so what cell then is going to come along and scavenge and phagocytose these hyaline bodies and then move along its way? It's a macrophage. So it's basically the garbage collector. It's a way you can think about that cell. Remember, macrophage. Macro means big, and phage means eat. So it's a big eater. Just eats all kinds of things. The cleanup cell. All right, so it will consume those hyaline bodies and move on its way. All right, here's a question for you. So apoptosis, does it require energy? The answer is yes. And what is the form of energy? It is an ATP. And actually there's a nice way to remember that. So the word apoptosis begins with an A, as does the word ATP. And I mean, in general, ATP is like the common currency for energy. It's used in just about every reaction. So let's now talk about the two main types of apoptosis: intrinsic and extrinsic. And the real difference between these two is just where the signal for apoptosis begins. So if it originates within the cell, then that would be intrinsic because it's within the cell. If it originates outside of the cell, that is extrinsic. It's as simple as that. So whatever signal is telling the cell to kill itself, if it's inside the cell, it's intrinsic. If it's extrinsic, then it's outside the cell. The first example that we'll talk about is in embryology. So when an infant is born, normally the fingers are separated actually through apoptosis. They were all joined together like we see here, but in the womb, apoptosis takes place and the fingers are separated. So when there's failure of separation and coming together of fingers, what's that term called? Syndactyly. So he's going to need some surgery. All right, we can also see intrinsic apoptosis in hormonal stimulation and withdrawal. So normally, what two hormones stimulate the uterine lining to proliferate and hang around in a woman? It's estrogen and progesterone. Okay, very good. And so if these are removed, then the uterine lining will be shed, and that's what occurs once a month through menstruation. And menstruation occurs through apoptosis. A third example of intrinsic apoptosis is in retired cells like neutrophils that have been around a site of inflammation long enough. After about 48 hours, they start to fade away. And they leave actually through apoptosis. The fourth and final example of intrinsic apoptosis is trauma. So if there is sufficient radiation like we see here or toxin exposure, the cell is just going to be damaged enough to the point where it knows that it can't continue on anymore and still be a functional contributing cell. So it will just basically off itself and tell the rest of the cells, "Oh, I can't make it anymore. Go on without me." And then it will die through apoptosis. Okay, so now that we talked about intrinsic apoptosis when the signal originates on the inside, now we're going to talk about extrinsic apoptosis when the signal originates on the outside. And really there's two main examples of extrinsic apoptosis, and both of those examples are just two types of cells. We're going to talk about natural killer cells, and then the next one we're going to talk about cytotoxic T cells. Both these cells tell other cells that they need to undergo apoptosis. And what types of cells generally will these be telling? So I'm asking you, what do you think? So in general, virally infected cells, or what else? Cancer cells. Basically, the types of cells that are defective and have something wrong with them. They need to go. And these immune cells will tell them to. Alright, so we'll briefly review immunology and natural killer cells. So they're natural killers. Here's a cell membrane here. And let's say a cell is infected with a virus. Ooh, that's not good. So normally, proteasomes within the cell will actually degrade the virus into fragments. These fragments will be inserted into the rough ER, rough endoplasmic reticulum, and from there they will be attached to a receptor on the surface. What type of receptor do you think they will be attached to? Answer is MHC1. Now, it has to travel from the rough ER to the surface. How is it going to get there? It actually arrives to the surface and is transported by a fragment of the MHC1 receptor called, what do you know? It's the beta 2 fraction. And actually, this is important on the USMLE. We'll discuss an example of why in a little bit. But anyway, we have a fragment of the virus on the surface. And this allows the natural killer cells to come and make contact with the receptor, recognize that it's infected with a virus, and say, all right, this cell needs to die. It's a danger to the whole body. And so what it will do is actually perforate a hole using its protein perforin. That makes sense. That is appropriately named. And after it's perforated a hole, it will deposit, what type of enzymes do you think it will deposit? Granzymes. Granzymes go in and actually kick off the apoptosis cascade. So they're going to go, what we just talked about, what's going to be released from the mitochondria. Cytochrome C, okay, good. And then that's going to go stimulate caspase enzymes, which will then trigger apoptosis and destruction of the cell in a nice, neat, organized fashion without any inflammation whatsoever. We'll contrast that with necrosis later. Okay, what about cytotoxic T cells? So here is another virally infected cell. It needs to go. So a cytotoxic T cell comes along. Remember, the receptor is CD8. Okay, and there's typically two stimulatory signals that are necessary for an immune cell to be fully activated. The first one is these two receptors here and here. What are those? What do you think? So it's fast ligand on the CD8 T cell, and then what on the virally infected cell? The fast receptor, another name for that is CD95. Okay, what's the second signal? That would be the release of something from a helper T cell, a certain interleukin. What do you think? Interleukin, it's the second signal. It is interleukin 2. So that will go and stimulate the CD8 cell to trigger the destruction of this virally infected cell through apoptosis. Again, cytochrome C from the mitochondrion activates caspase enzymes, and this cell is toast through apoptosis. Alright, so that was a nice little immunology review. Let's keep moving on here, and whoa, another flash quiz, wow. Alright, so besides apoptosis, what is the other main way that cells can die after sufficient injury? I mean, this is an easy one. You already told me earlier. So we talked about apoptosis. The next thing we will talk about now, next way cells can die, is through necrosis. And again,